Okay, I'm pleased that everyone could come to our Pharmaceutical and Personal Care Products Consortium meeting today. Those who don't, I'm Assistant Director here at ISTC and organize these consortium meetings along with Beth Machewski, who works with me. I want to first say we have a sign-up sheet that we're going to be passing around just so you can just check off your name if we've had you here attending before, and if you're new on the second page, you can just add your name and email information. That way we can keep you on the contact list, and we'll send that out to other participants here, so in case you want to communicate together, you can do that more easily. So with that, so this meeting gives us an opportunity to hear about each other's research, discuss ideas, and possible collaborations. Since we have some new people here today, I'd like to just take a minute to go around the room, and if you could stand up and say your name and then maybe a few words about your interest in pharmaceuticals or contaminants in the environment. So we'll start with you, Beth.
Hi, I'm Beth Machewski. I'm a researcher here at ISTC, and I work with Nancy, and I'm also working with Illinois Indiana Sea Grant on one of their projects.
I'm Christine. I'm not a researcher. I'm actually a journalist, and I'm here just because I'm really interested in this topic and I want to learn more about it.
I'm Walt Kelly, the head of the groundwater section at the Water Survey, and I've done some research with Way and Laurel on PPCPs in the environment, mainly in karst environments.
Lance Scheidemann, and I work here as a senior researcher at ISTC and do some work on engineered processes that help remove PPCPs.
Hi, I'm Peter Liu. I'm not a researcher. I'm just interested in this topic. I saw it on a calendar, so I decided to come.
Hi, Jeff Levengood. I'm a research scientist in fish and wildlife toxicology with the Illinois Natural History Survey, and I've dipped my toes into the PPCP waters.
Hi, I'm Laura Kamen. I'm the Pollution Prevention Program Specialist with Illinois Indiana Sea Grant, and Illinois Indiana Sea Grant is working with ISTC on a PPCP project currently, and since we do outreach, I just came to learn more so we can share that.
Hi, I'm Terry Hallisey, also with Illinois Indiana Sea Grant, Education Coordinator, and Laura mentioned our role.
I'm Kirsten Walker. I'm also with Illinois Indiana Sea Grant, and I just finished creating some new curriculum for the medicine test, which is focused on PPCP research.
John Scott, ISTC Senior Analytical Chemist. My laboratory does a lot of the measurements and methods developments for PPCPs.
Mike Machewski, State Water Survey. I've done work with them in the past with people like Way and John, but nothing current, but just trying to keep up on the topic.
My name is Abigail Bobber, and I also work for Illinois Indiana Sea Grant as a communications specialist.
Adrian Gully, and I work with Illinois Indiana Sea Grant. I'm the outreach specialist, so I educate communities on the PPCP projects.
My name is Wei Zheng. I'm research scientist at the ISTC. I work on the PPCP, including the PPCP's environment, fate, and transport, also for the mitigation strategy. So most of my research focus on the research parts. Then Nancy and Laura, they are for the Sea Grant. They work on the outreach and extension parts.
I'm Laurel Dodgen. I'm a postdoc with Way looking at PPCPs and other organic contaminants in reclaimed water and other kinds of water sources.
My name is David Noreen, and my area is experimental psychology and statistics. I've taught that in the psychology department for quite a while, but I got interested in this through Claudia Linhoff giving a nice talk a few years back on nanoparticles in different products that were getting into the environment. And then I've also been looking at the Chanute Air Force Base where there's an agriculture program that initially planted their first crop in an area that was badly contaminated with PFCs. So I've been following that project and very interested in PFCs and emerging contaminants.
Hi, I'm Jackie Christensen. I'm Director of Operations for the Urbana and Champaign Sanitary District. Obviously, we do wastewater treatment. I'm trying to follow the research and see what potential impacts it may have for us in the future as a utility, and basically on the type of treatment that we might have to implement in the future. I know we've provided some samples for some of the researchers here in the room. We did get some results back and just was interested in seeing the concentrations coming in and what is actually being discharged. So sort of a preemptive learning before things get into the regulatory stage. I'll probably be long gone before that, but I like getting in on the groundwork.
Hi, I'm Madeline Van Middlesworth. I'm a graduate student here at U of I for the Illinois River Biological Station. We're currently researching different measures of endocrine disruption between fish species in our Illinois River.
Great, thank you everyone. And we may have a couple of people online on our speakers both today. As you may have seen, we'll be doing webinars from Minnesota and then from Southern Illinois. So we'll get to our speakers in just a moment. I did want to say that for our spring meeting for our consortium group, we will just have that be part of our April 4th PPCP conference that we're having at the iHotel. And so with our announcement on the consortium, you should have seen the conference call for abstracts, and we'll be keeping you informed about the program for that coming up. But we thought you could all get together there and discuss further our work, and hopefully we'll have lots of researchers from different parts of the country there also, so we can hear more about all of the work going on with pharmaceuticals and personal care products and other emerging contaminants in the environment. So mark April 4th down, and you'll be getting lots of notices from us about the conference. So with that, as we begin the two talks today, I'd like to ask everyone in attendance to please silence your cell phones. And then we're going to hold the questions to the end. The speakers will each be giving about a 20-minute presentation about their programs, and then we can ask the questions at the end. I'll come around with the microphone. It does not amplify here in the room, but we're doing this because we are recording this. And we'll have the both of the talks available online on our website. I'll send out the link afterwards in case you have people who weren't able to attend today who would like to hear these talks or you want to review what they said. But the microphone is, I'll pass around for any questions so that we can record those. So that's why we're speaking into this. So with that, our first speaker will be Dr. Helen Godin of the Minnesota Department of Health. She's going to discuss the Minnesota Department of Health's drinking water contaminants of emerging concern program. Dr. Godin is a senior toxicologist and human health risk researcher for the Health Risk Assessment Unit at Minnesota Department of Health. She's currently the scientific lead for the Drinking Water Contaminants of Emerging Concern program there. Her responsibilities include toxicologic assessment of a wide range of environmental contaminants, for example, industrial, agriculture, pharmaceutical, and consumer products, the development of the statewide health-based criteria for groundwater and drinking water. Has a leadership federal work groups regarding development, improvement, integration of risk assessment methods, and public health policies that are protective of sensitive or highly exposed populations, for example, infants and children, and case-by-case health risk assessments or research projects specific to emerging environmental health threats, for example, perfluoric chemicals. Dr. Godin has served as a member of the EPA's Chemical Assessment Advisory Committee, several EPA chemical reviews, and currently serves as a member of the NSF International Health Advisory Board. She's lectured on toxicology and risk assessment at the University of Minnesota Schools of Public Health. She's a member of the Society of Toxicology and a founding member of the National Dose Response Specialty Section of the Society for Risk Assessment. Dr. Godin received her PhD in Environmental Health and Toxicology at the University of Cincinnati. With that, I'll turn it over to Dr. Godin, and we'll begin her presentation.
All right, thank you, Nancy. Can you hear me all right? Yes, we can.
Okay. And I'm assuming you can see the slides okay?
Yes, they're coming through nicely. Thank you.
All right, great, thanks. Well, I was very pleased to receive the invitation from Nancy. We're always thrilled to share our work and also to talk with peers who are also involved in emerging contaminants. So as many of you know, there are several federal agencies in the United States that are involved in emerging contaminant work, and within the state of Minnesota, there's also several state agencies as well as academic institutions that are involved in this work. And today I'm speaking really only just on one program within the Minnesota Department of Health. Just very briefly, I'm going to give you a little bit of background on our Clean Water Fund activities, very briefly, and then share with you sort of our three-pronged approach within our program. So chemical or contaminant evaluation process, some of our research activities, and then outreach and education. First of all, I wanted to share and acknowledge where our funding for our program comes from. In November of 2008, voters approved a Minnesota constitutional amendment that resulted in a sales tax increase to raise money for several funds. One of those was the Clean Water Fund. And this fund, part of it was dedicated strictly to drinking water protection, and it's out of that portion that funds our program. At the Minnesota Department of Health, water-related activities include protection of source water, sealing unused and abandoned wells, expansion of our county well index database, and then finally the drinking water contaminants of emerging concern program that the rest of my talk will focus on. So why was there why was our program created? Why was there this interest in emerging contaminants? What are they and what's the concern? Well, as most of you in the audience are fully aware of, we live in a chemical society. We use millions of tons of chemicals in industrial uses, agriculture, consumer products. So it's not surprising that these chemicals from products that we use, et cetera, find their way into our environment, and the environment has become a sort of a reflection of our lifestyle. And we also have some very good chemists who are very curious and are out there sampling in many places, including people's bodies, and finding contaminants. So, of course, when they're found, particularly when they're found in sources of drinking water, this will ultimately raise questions. In particular, is there a risk from this exposure? Unfortunately, sometimes the answers aren't readily available. Very few of the contaminants that there's concerns about have health-based guidance values. For example, EPA has maximum contaminant levels, which are the standards used for public water systems, for only about 90 compounds, and they have health advisories for under 200 additional chemicals. So there's not very many health-based guidance values versus thousands of contaminants that may be out in the environment. So the fact that we can't give an answer often, this results in people becoming fearful, concerned, and there could be inappropriate reactions to this. So one of the impetus for our program was to try to provide context to answer some of these questions. We started with defining what we meant by contaminant of emerging concern. There are many different definitions, and they don't necessarily agree with each other, but this is the definition that we came up with. It's very broad. We defined CECs as substances that have been released to, found in, or have the potential to enter Minnesota waters. And they may pose a real or perceived health threat. They do not have Minnesota health-based guidance, or there's new or changing health and exposure information that would increase the level of concern, and thereby our previously existing health-based guidance may no longer be considered adequate. So as I indicated earlier, we have sort of a three-pronged approach. One is chemical evaluation, and we do two levels of evaluation. One is a screening level review, and then one is an in-depth full review, which ultimately results in typically numerical human health-based guidance values. We also have efforts in the area of research specifically to try to improve our ability to provide risk characterization and improve and provide risk communication. And then lastly, outreach and education. Currently, our program has roughly about 6 staff that devote part or most of their time toward this effort. I also want to mention that this is a new initiative for our department. It allows us to expand our work and proactively address a wide range of chemicals before they're detected in drinking water. Prior to this program, our efforts really focused on supporting our sister agencies in remediation programs, and so it really focused on more of the legacy contaminants and reacting after contaminants had been found impacting largely groundwater. That program still does exist, and it's one of our sister programs we work closely with, but this Emerging Contaminant Program is new. I apologize, this is a very busy slide. It's the busiest slide I have in my presentation, but I wanted to sort of show you the process of our chemical evaluation from start to finish. So it starts with a nomination, and there is a web link there. Anyone can nominate a chemical. So far, we've had about 90 chemicals that have been nominated for consideration, and a very wide variety: pesticides, pharmaceuticals, personal care products. The vast majority of those have met our definition. Only about 10 have not, and those were chemicals that were already under consideration or being dealt with by our sister program. So the screening assessment is a very quick gathering of available toxicity and exposure information with a focus on the potential to impact drinking water sources. And we use this information, and the chemicals are assigned a score and they're ranked, and those that are ranked higher are evaluated or tapped for full review if possible. There are some times that we do not have enough information to even score a chemical, let alone develop a full guidance value for it. And I just want to clarify too that this ranking is relative to other screened chemical. It's not the entire universe of chemicals. For our guidance development, we do an in-depth evaluation of both the toxicity and the exposure information using standard risk assessment methods for deriving drinking water values. And typically we complete 4 to 5 in-depth reviews per year. And to date we've completed guidance for about 28 chemicals. For outreach, we develop informational materials, and I'll show you an example later on in the presentation. These materials are both technical as well as general in nature. And the web link at the bottom of the slide actually is to what we refer to as our nominated chemical status table. It's a evergreen document for any chemical that's been nominated. We, in a very concise way, summarize the information that was provided by the nominee and the status of that chemical within the chemical evaluation process. That table we do try to update on a quarterly basis. So I encourage you, if you're interested, to check that out. This is just a very simple diagram reminding me to sort of qualify. We are typically interested in the chemicals that end up in that upper right quadrant. That indicated they have higher toxicity and exposure concern, and therefore a higher risk concern. But we also do select, on occasion, chemicals that fall outside that quadrant, depending on program needs for other sister agencies. And for one example I can give you is isobutanol, which did not rank high on the level of concern ranking, but some of the ethanol plants in southern Minnesota were converting to isobutanol production, and our sister program needed guidance for regulatory and permitting purposes, and none existed. And so we stepped up and developed some guidance values for that purpose. This is a list of the 28 chemicals I mentioned that we've completed. I'm not going to read the names to you, but you may see chemicals there that may be of interest to you. Each of those do have what's referred to as a technical summary, which outlines exactly how our guidance values were developed for that chemical, and as well as a general information sheet, and I will show you an example in the next slide. Currently, we have 4 chemicals that are under review, and we do anticipate that those reviews will be completed in the next 4 to 6 months. We also have a list of contaminants that are evaluated with the sister program I mentioned to you, and so there are many more guidance values available than just the list of 28 that are presented here. This is an example of what we've referred to as an information sheet. This example is on triclosan. This is intended for the interested public to provide general information on the chemical. It also allows us an opportunity to provide recommendations to the public on how to reduce their exposures and also how to reduce release to the environment, which is particularly relevant for consumer products. So you probably cannot see in this slide, but within the materials there's a clear recommendation from the Department of Health that we do not recommend using triclosan-containing products unless people are specifically directed to do so by a healthcare professional. So we do take the opportunity to make these kinds of statements to the general public. And as I indicated, there's also an accompanying toxicity summary sheet that goes along this for triclosan, which shows for multiple durations of exposure, our points of departure, what kind of uncertainty factors we've applied, what reference dose we've derived, and the health protective water guidance value for the different durations. We have a whole separate technical document that provides our methods of how we do this. We're unique in that we do develop, if possible, guidance for short-term, subchronic, and chronic durations of exposure, which we feel is extremely important for drinking water, since short-term exposures, particularly in bottle-fed infants, are much higher on a per body weight basis than adult consumers. Next, I'd like just to highlight some of our research activities. So in addition to sitting at our desk looking over toxicity information, we do have additional activities that we get involved with. So first of all, we do fund staff and equipment at our public health laboratory here at the Department of Health. We utilize that staff as a resource person in terms of environmental fate and chemistry. We also work with them to identify needs for development of analytical methods. So for example, in the last year or so, staff there that we support have developed and published analytical methods for acrylamide, a series of estrogens, as well as benzotriazoles. And we're currently working with them looking at some additional methods developments for some pharmaceuticals. Our alternative risk assessment method, this really is aimed at those chemicals for which there's little or no toxicity information specific to that chemical, and unfortunately this is the case for many chemicals. We understand it's very unsatisfactory to tell someone, well, yes, we have found this in your drinking water, but we have no idea what that means for you. So since that's, like I said, not a very satisfactory position to be in for anyone, we are evaluating tools or methods by which we may be able to provide at least some risk context for when we discover those types of chemicals. And this is an ongoing project. We have completed a couple phases within the project, and those are executive summaries from those reports are on our website, as well as a couple of posters that we have presented at Society of Toxicology. Another project is the naturally occurring radionuclides in groundwater. As some of you may know, there's a variety of radionuclides that can be responsible for gross alpha particle activity, but they vary widely in their potency, their level of toxicity. So this was a pilot project to identify specific radionuclides that were responsible for elevated gross alpha particle activity in specific public water supplies. And that pilot project has been completed, and a report is being finalized and hopefully will be posted to our website very shortly. Pharmaceuticals and pesticides are among the only groups of chemicals for which oral toxicity testing is actually required by federal agencies. And these federal agencies review and summarize this information. And so these two types of chemicals sort of represent low-hanging fruit to us, and we divide two projects to leverage this kind of information. So within the projects, methods were developed to allow for a very rapid derivation of health protective benchmarks, and this was done for about 120 active pharmaceutical ingredients. We targeted the active ingredients that were most frequently prescribed within the United States, in addition for about 160 pesticides. Now, these benchmarks are very useful in providing some risk context, and they've also been very helpful to prioritize which pharmaceuticals and pesticides may warrant that full in-depth evaluation. Both of these projects, at least the initial parts of them, have been completed, and the reports have both been posted on our website as well, which the web link is at the bottom of the page. We also have a microbial risk assessment project that's in process. There are two reuse systems that we are studying in particular, looking at how various conditions such as intense rainfall or extremely hot weather may change water quality. At the time when we can determine which pathogens are present and quantify them, we do plan to actually conduct a quantitative risk assessment on this work as well. And the last project I wanted to mention was a database of USGS Minnesota water occurrence data. As many of you are aware, USGS is a very key agency that collects contaminants of emerging concern occurrence data. And to facilitate our work, we partnered with them to create a searchable database that contained Minnesota water monitoring data. And our staff are currently beta testing and conducting searches on the database, and it looks very promising, and it would greatly enhance our ability to quickly conduct searches. And all of this information is available. More information, more description of these projects are available on our special projects web link, which is again noted at the bottom of the slide. And on the outreach and grant. Variety of values are quantifying risk to provide risk context, but they actually address the upstream issue of how do these contaminants get into the environment in the first place. And so education is really a key part of our efforts. So we have provided grant and funding to create and participate in community events to generate new literature and media projects that increase the awareness and understanding of emerging contaminants in drinking water, as well as personal action that can reduce these types of contaminants. Video that is just an example of some of the media campaigns to sort of heighten awareness of what people can do to decrease their sort of footprint. And to date, we've awarded grants that have a total of $200,000. So at the beginning of the talk, I indicated that one impetus for the program was to provide context regarding CECs in drinking water. So on the next few slides, I just want to show you one example of what we've been able to do in this regard. Last summer, the USGS and the Pollution Control Agency published the results of a study of emerging contaminants in the ambient groundwater in Minnesota. The groundwater serves as a source of drinking water for the majority of Minnesotans. And this report indicated that low levels of at least 35 contaminants were detected in just ambient groundwater. This is groundwater where they focused on areas away from any point source that they were aware of. So needless to say, this raised questions and concerns of citizens of Minnesota, which of course was, well, should we be concerned about this? And this shouldn't be in our drinking water. What are you doing about it? So one of the first questions was, well, can we provide some context? So out of that list, there are 4 EPA maximum contaminant levels or health advisory values. So that left about 90% of the detected contaminants without any type of comparison for health-based guidance. So Minnesota Department of Health had full review guidance for about 17 contaminants, and some of these guidance values were developed by our sister program, looks more at the legacy contaminants. Through the rapid assessment, we were able to provide benchmarks for additional 6 contaminants, and then with some of the alternative tools, we provide some risk context for another 7 contaminants. So this resulted in being able to provide some level of risk context for nearly 90% of the contaminants, which was sort of the flip side of the availability of federal guidance values. So and in addition to the guidance and benchmark values, and frequency of detection levels have been used to prioritize contaminants, again, for efforts for more in-depth reviews and, in some cases, analytical method improvements. So quickly, just to summarize, our program, which is funded through the Clean Water, Land, and Legacy Amendment, does provide risk context for contaminants of emerging concern that frequently have no health-based water guidance at the state or the federal level. We have provided research that can improve and create additional approaches for assessing potential human health risks, and it's a particular area that we are still actively working in. And we also provide public outreach and education to increase the awareness of how personal actions can reduce exposure and environmental impacts, because we really feel that this is really the long-term solution to the situation. So that ends my talk. I wanted to leave plenty of time for questions. My contact information is here. There are some of the key pages. The little spotlight icon with that little rose-colored envelope is a reminder to me to let you know that if you go to the home page, in the right-hand bar, you will see under Spotlight the little rose-colored envelope. And you can click on that and sign up to subscribe to our notification system. If you're interested in tracking any of our activities, we periodically will send out updates to our subscribers. We do not inundate you with updates. You know, there may be a couple of updates per month. But I actively encourage, if you're interested in following any of our activities, to sign up. And also, if you are interested in nominating any chemicals to our program, please feel free to do that. So that completes my presentation. I'd be happy to answer any questions or provide more in-depth information on any of our activities. Thank you.
Okay, thank you very much, Helen, for your very good summary of all of your programs there. So I'm going to ask the audience here now if they have any questions, and I'll pass the microphone around so we can record those. Are there questions from the audience for Helen?
Hi, Helen. This is Laura Kamen from Illinois Indiana Sea Grant. Thank you. That was a very good talk. My question is, for chemicals that are reported to you to request assessment, how many chemicals are you able to assess each year? Do you have sort of a target number?
Actually, we do have targets. Our funding mechanism requires us to have very specific matrix, and they check with us every year to see if we're doing our job. So as I indicated, those full, very in-depth reviews, we do about 5 per year. But our screening assessments, we typically do at least 20 per year. And, you know, part of that depends on the chemical. Some, if you can't find information, obviously it's a very quick assessment. And one of the things we have started to do is we've started to create small, what we call screening profiles, just like one sheet of paper that provides at least some summary of the information we've been able to gather at this screening stage. And to date, we have been able to screen all of the chemicals that have been nominated. We've just gotten a nomination of about 15 additional chemicals from our sister agency in August, and we're about halfway through those. So hopefully that gives you some idea of what we're able to do. Obviously, if we got nomination of hundreds of chemicals, it would take us a while to get through those.
Okay, thank you, Helen. Are there other questions here?
Hi, Alan. This is Wei. I have a question. As you know, the Minnesota, like Illinois, I think you guys should have a large animal farm. Do you target, or did you target some like a pharmaceutical from the like a large animal farm, like a veterinary pharmaceutical, veterinary antibiotics, or like animal hormones?
We focused on what's prescribed for humans, and so obviously there's some overlap in between human and animal use. One of the areas the project that we looked at, as I indicated in the talk, was we tried to leverage the information that's out there for the active ingredient. So what we focused on was the detailed drug labels from FDA, which you do not have for some of the livestock types of veterinary use drugs. So as I indicated, what we did is sort of the low-hanging fruit. Certainly the volume of medications that are used in livestock production and treatment is huge. So this is an area too that we would take a closer look at, and some of the actual contaminants that were submitted by our sister agency do include a couple of livestock types of antimicrobial or growth enhancement type of compounds. So if the information is available to us, we can look at those and try to develop some type of guidance. To date, we've only done a couple of those. So it really does depend on the availability of the information.
Are there any other questions here from the audience?
Yeah, this is Lance Scheidemann from ISTC. I had a question on your microbial risk assessment model. If you could describe maybe some of the inputs or the outputs of that model. Just curious what it does and what it needs to do that.
Well, as I'm not the actual lead for that one, so I would have to, if you could send me your contact information, and Nancy here is focusing on that. We are partnering with the University of Minnesota, looking at who are doing the two reuse scenarios that we're looking at. Is one is irrigation of a recreational area, and another is non-potable uses within a dormitory at the University of Minnesota. And Nancy has been developing the quantitative model, which to my understanding we have not run at this moment yet because they're still trying with the water use systems that they're evaluating. But Nancy does have a model and has and she could tell you the specific inputs, et cetera, of what she needs to run those models. So I'm sorry, I can't answer that question directly, but but one of our staff people can.
Okay, thank you. Other questions?
Hi, David Noreen. I'm interested in PFC contamination. I didn't see that in your chart, but I've read a little bit about how with very sensitive detection mechanisms it's been detected in the water supply of Minneapolis-St. Paul. And I was wondering if there's been any hotspots that have been found in Minnesota, like around 3M, where they were manufacturing products with PFCs?
Yes, it's probably old news. We had quite massive contamination from disposal areas from 3M, and that sort of all exploded back in 2002 and 2003. So we have number of private wells that are on treatment systems, as well as one or two very large public water systems that have treatment on to remove perfluorinated chemicals from the drinking water. Though that our program, the Emerging Contaminant Program, was only initiated in 2009, so that most of that PFC work preceded the creation of our program. But I believe, those types of episodes, because PFCs were an example of where there was almost no published data available for the contaminants, and yet there was obviously a very high need to provide some kind of risk context for what was being found. So I think it may have been one of the impetuses for funding the type of program that we currently have. So there are definitely perfluorinated chemicals still in the environment here, and the treatment is still underway. And one of the, I don't know, I may have gone through it too quickly, one of the contaminants that we are currently doing a review on is PFHXS, which is one of the perfluorinated chemicals. We're actually looking at using some of the alternative methods to determine whether or not we can do some kind of extrapolation from other PFCs where there's very little chemical-specific toxicity information on still many of the perfluorinated compounds.
Okay, thank you very much, Helen. Is there one last question here at all? Okay, with that, we will move on and switch to our next presenter. So thank you again, Helen.